shorter, they could be regular or irregular, but whatever it is, a normal woman would have these regular or irregular menstrual cycles. And if they are not there, then either the woman, they are not there, it could be two reasons. One, of course, they have not initiated, so they haven't attained menarche, or if it happens after they attain menarche, either they have reached menopause early, or there could be some other reason. So if you don't attain menarche, then you are looking at primary amenorrhea. If you, as I said, if the cessation of menarche happens early, it could be secondary amenorrhea or a premature ovarian failure. Now, the underlying that secondary amenorrhea could be premature ovarian failure. Now, if you look at a definition for primary amenorrhea, it can be defined as the absence of menarche by the age of 14 without secondary sexual characteristics, and or if there are secondary sexual characteristics, absence of menarche by the age of 16 years. So in genetic terms, we define it for genetic evaluation. Secondary amenorrhea, on the other hand, is the absence of menses for more than 6 months or the duration of 3 menstrual cycles. That could trigger a genetic evaluation. Now, if you look at the causes of primary amenorrhea, the chromosomal abnormalities would be the main reason. So those chromosomal abnormalities would result in gonadal dysgenesis. Then there is hypothalamic hypogonadism, which is accounting for about 20% of the cases. And then, of course, the anatomical stuff like absence of uterus, cervix, or vagina. That's also there. And there could be other anatomical issues as well: vaginal septa, imperforate anus, and imperforate hymen, and so on. And then, of course, finally, the pituitary disease. So all of these should be in your mind. And why I said that primary amenorrhea should be trigger the thought of a genetic referral would be obvious to you when you look at this, because the majority of the cases would be due to chromosomal abnormalities, and therefore it is important to have a genetic evaluation. Now, just to remind you, let's go back to the first year where we talked about the determination of the sex of the fetus at the time of fertilization. We know you get an X chromosome from the mother, X chromosome from the father, and you end up becoming a female. If you get an X chromosome from the mother and a Y chromosome from the father, you end up becoming a male. And as you remember, the sexual differentiation begins when the primordial germ cells arrive at the intermediate mesodermal region where the gonadal ridge is there from the yolk sac. So you may remember this slide where we saw the migration of the primordial germ cells from the yolk sac all the way and arriving at the gonadal ridge where around the 6th week of intrauterine development, and eventually those primordial germ cells, arrival of the primordial germ cells, triggering a cascade of events which eventually leads to the formation of the ovaries and the testes. Now, to again remember, what we begin is with an indifferent gonad, and it would the natural tendency, if you want to call it, is towards development of towards an ovary. But when a Y chromosome is there, the SRY gene results in the production of the testicular determining factor, which converts these indifferent gonads towards a phenotype of a testis. And once the testis is formed, it produces testosterone, and the testosterone goes and binds its receptor in the various tissues, bringing about the differentiation of the somatic sex of the individual. So the receptor which is in the cells, the androgen receptor to which testosterone binds, is determined or coded for by a gene in the X chromosome, the androgen receptor gene in the X chromosome. So the entire sexual differentiation, whether you are a male or a female, requires the presence of both of these genes, the SRY gene, which produces the testicular determining factor, the androgen receptor gene, which determines the production of the androgen receptor. So going a little bit further, now once the testosterone enters the cells, it gets converted to dihydroxytestosterone by 5-alpha reductase. Both of these can bind the androgen receptor. So if testosterone binds, when the testosterone binds the androgen receptor in the duct system in the developing embryo, the male mesonephric ducts, development of the mesonephric duct system is promoted. And when the testosterone binds the 5-alpha dihydroxytestosterone binds the testosterone androgen receptor, it promotes the development of the male external genitalia. So the entire complex process has to work towards the development of the male phenotype, as it were. In the absence of testosterone in a female, then these differentiations wouldn't occur, and as a result of that, the phenotypic female would, the phenotype of the female would manifest in the person. All right. So the key message here is that both the testosterone and the testosterone receptor, androgen receptor, have to work. If there is a mutation in the androgen receptor and the testosterone or the dihydroxytestosterone cannot bind, then what happens? Then what happens is this. Then what happens is the male phenotype wouldn't manifest. So you will remain, even though you have the Y chromosome producing testosterone in your, Y chromosome producing a testis in your body, and that testis secreting testosterone, you would still end up with a female phenotype. And we know what the condition is. We'll see that in a little while. So to summarize this again, presence of the Y chromosomes, you get the SRY gene, which produces testicular determining factor, and it acts on the indifferent gonad and makes it a testis. Whereas in the absence of these, it will become the indifferent gonad would be an ovary. And you can remember from your first year's work what happens in that differentiation. I'm not going to go into details of that here. If you're interested, draw out Langman and take a look at it. The testis, as we said, produces the androgens or testosterone, as well as another substance that you remember, the Mullerian inhibitory substance. So it suppresses the paramesonephric system or the Mullerian system and promotes the development of the mesonephric system, which is the male duct system. So we have the both the genital ducts and the external genitalia formation because of this hormonal influence. In the females, we in the male, of course, in the females we get estrogens, and in the presence of estrogen, the female phenotype manifests. And then, when this occurs, we say that the genital sex is established. So we have the with our genes, we have the determination of the genetic sex. But if all the entire system works properly, then we end up with acquiring the genital sex that we are supposed to have, right? So that's what we would get. Now, of course, you can go back, read about these, and also look at the first term stuff where we looked at what happens to these different structures. I'm not going to go into details of that, and then eventually, of course, you will have the definitive organs which are produced. And again, if you go back to your first term stuff, you will see what happens with these different, what are the different features that happen in the external genitalia as well. And you would remember some of the stuff that we talked about on formation of the scrotum and penis and so on, as well as the external genitalia of the female. So just go back to your first year stuff and refresh your minds. Now, once the genital sex is established, there's further hormonal influence, development of secondary sexual characteristics, and so on, and then the somatic sex is formed. So it's not just about the genitalia, the determination of the sex. It's also the whole appearance of the individual. In a woman, secondary breast development, and so on as well. So with all of those, you will have a secondary sexual characteristics developing and establishing the somatic sex of the person. And underlying this somatic sex at genetic level, we would have the we can have XY males, and then that would be the normal. Then of course there can be XX males as well. Then of course the other forms like XXY or even a higher multiples of these are there. As long as there's a Y, you will generally have a male phenotype, and we'll come back to see how we have a XX male later on. Now, looking at the females, so the females will have a XX female phenotype, but then you will have a XY females as well. That is when there is a mutation in the SRY gene, mutation in the androgen receptor gene, where we know that it is androgen insensitivity syndrome. And then, of course, those who have only one X chromosome, which is the Turner syndrome. So you can see underlying their somatic sex, we have to keep in mind that these genetic etiologies could be there if you have a person, a woman presenting with primary amenorrhea. Then, of course, eventually, based on other influences, you will have a social sex, which is developed. There may be environmental factors, sex of rearing, gender, all kinds of other things can come into play, and you may assume a different role in your social sex. It could be the same as the somatic sex, but then of course there are others where there's diversity within our population. There is the LGBT community and so on. So you may assume a different social sex. And we have to accept that within the limits of diversity that we see in society. Right. So moving on, when we look at clinical presentations, if this whole thing does not happen properly, or the development does not happen properly, you may have seen when you are doing your pediatric appointments, children with ambiguous genitalia. But they will be picked up very early, and it's rarely that they will be present with ambiguous genitalia to gynecology wards. It will be very rarely that somebody is missed and will be missed for so long. However, what we get is if this process doesn't happen properly, there's underlying genetic defect, then that is primary amenorrhea is the one which you will see. Then, of course, rarely you may also see females who are masculinized women would be there, and we'll see why they are. Then, of course, there can be even under masculinized males. So, in the context of androgen insensitivity syndrome, this entire spectrum can be there, and I'll explain that a little bit in a little while. Both not only primary amenorrhea, but also the other ones like under masculinized males can also be there. Then, of course, you can have other presentations like males with abnormal breast development may come, but of course they won't come to gynecology clinics. They may be seen mainly in medical or endocrinology clinics, where you have Klinefelter syndrome, males with breast development who may come. Then, of course, short stature can also be there with Turner syndrome, and they may come as as children who are not growing tall. Then, of course, they may present as with primary amenorrhea and so on. But the short stature could be an additional feature to primary amenorrhea that you may that may trigger you to think of Turner syndrome. Well, the other features of Turner syndrome, like with neck and so on. Those may not be obvious sometimes, but short stature, of course, would always be there. So short female with primary amenorrhea, you must be thinking of Turner syndrome if they are coming to a gynae clinic, right? So, but you must remember, amenorrhea is a symptom; it's not a disease. So, therefore, you need to go through. You must go through the family history. So, once you've taken the usual history, you must go into a little bit of detail of family history to find out whether there are any more people in the family with primary amenorrhea. So, so why? Because if it's androgen insensitivity syndrome, there would be people, because it's X-linked recessive, there would be people with primary amenorrhea in that family. There can be, uncles and there can be aunts of this particular woman who may be subfertile, who have not had children in spite of getting married. There could be, because this, as I said, the androgen insensitivity syndrome is a spectrum. In the family history, there could be even males who are married but who don't have children, and so those are the under-virilized males who may be there. Rarely those ones are also there in this family. So so looking at a complete subfertility history is important. It's not a it's probably not a primary amenorrhea history that you are looking at. You are maybe looking at a subfertility history in that family. So you have to ask not only for the presence of people who did not have a family, who did not menstruate, but also those who do not have children. That would, help you work out the pattern of inheritance if it's androgen insensitivity syndrome. Then, of course, moving on to examination, you will have to look at the, obviously, examine the breasts. You will have to do a pelvic examination to look at anatomical causes. And then you may go and do ultrasound scanning or laparoscopic testing. And then, of course, you will also do hormonal levels, FSH and LH. You may remember the hypothalamic pituitary ovarian axis lectures that I did, as well as you would have learned a lot from the more than what I did from the physiologists. And then maybe in your reproductive health module, somebody is reminding you of those again. And then, of course, genetic testing as well. So you need to look at all of these things: history, examination, investigations, and so on. And so this day and age, because genetic testing is available, you might, before you go into do invasive things like laparoscopic examinations, once you've got done the initial bits, done ultrasound scan, got a LHF, FSH levels done, you may go and do the genetic test. So you will remember from your first year's work the different anatomical causes that would contribute to a primary amenorrhea. So refer up those. That would be, of course, detected in by the time you do pelvic examination, and then do the laparoscopic examination. Do a ultrasound scan, probably get to somewhere there. But when you look at if you look at the presence of breast development as a main feature, there are things that you can, there's a framework on which you can, go a little bit further. So if you have, if the breast development is not there, but when you do the ultrasound scans and the pelvic examination, you see the presence of the uterus. Of course, must remember, in a, unmarried girl, you will not go and do a vaginal examination. You'll only do a visual examination of the pelvic region, right? So first things that you will do in this kind of patient would be just getting a FSH level done to see whether the hypothalamo-pituitary ovarian axis is working properly. So if the FSH, when you do the FSH levels, if the FSH levels are high, immediately then you are looking at a hypergonadotrophic hypogonadism. So that's what you're looking at. And then, of course, there's the gonads, probably there's gonadal dysgenesis, and you need to do a karyotype immediately. And then with that, you will detect the presence of the Turner syndrome. So that would lead you to Turner syndrome. And of course, there are various other forms in which Turner syndrome occurs. There are various other karyotypes you get with Turner syndrome. I've listed some of them here. Also, you can get mosaics like this as well. Then there can be higher, not only, so those are, these are some of the other karyotypes that you would get with Turner syndrome. Then, of course, let's go and show you a little bit more. So there could be others with things like this, where you have mosaicism. And then the other conditions which you get gonadal dysgenesis would be things like triplet woman with 3 X's, even something like this. All these are possible. And then going back to the other karyotypes I mentioned with Turner syndrome, you can see deletions of the chromosomes, long arms, short arms, and then you can also get things like this, dicentric chromosomes. So all of these things are possible, right? All of these different things are possible, and it's so when you do a karyotype, you will detect these things. Moving, so that is the most commonest diagnosis that you would arrive at. So Turner or its variants and higher multiples of the X chromosome are the ones that you would come up with in this group. However, you can also have a normal XX. And in that situation, you may be looking at any one of these. So you need to keep in mind, and you need to keep those in mind that would trigger the appropriate investigations. Now, that was when a breast was absent and the uterus was present. The second situation is there's breast development. So so in previous one there was breast development was not marked, but here there's breasts are present. There's a good breast development, but the uterus is absent. In this situation, you will immediately go to a karyotype, and if you get FOD6XY, you know that it is androgen insensitive syndrome. If it's FOD6XX, you may be looking at a Mullerian agenesis. So you can, before the karyotype results come, one way of suspecting these two is that in androgen insensitivity syndrome, there is no secondary sexual hair, that very sparse secondary sexual hair development, whereas in the Mullerian agenesis, there will be secondary sexual hair different, normal sexual hair. Why is that? Because of the presence of the normally functioning androgen receptor in the people who have Mullerian agenesis. Here, in this case, in the case of androgen syndrome, there's no normally functioning androgen receptor, and therefore there will be no development of secondary sexual hair. So getting on to androgen insensitivity syndrome, as you know, it's an X-linked recessive condition, mutation in the androgen receptor gene. And if you do their testosterone levels, you will find near normal male levels of testosterone would be there. And as I told you at the beginning, we see the interplay between the secretion of testosterone and then, of course, the production of the androgen receptor by the androgen receptor gene. So both those have to function properly. So a person, a woman with androgen syndrome will have a normal male karyotype. And if you look into a family, you may have a family like this, family history like this, where you have several members on the family who don't have children, who are subfertile, and if you do their karyotype, you will find all of them having a XY karyotype. Now, as I mentioned earlier, the androgen syndrome there can be a spectrum. There is complete androgen insensitive syndrome, and then, of course, partial. And also those with the male phenotype with these under-virilized males. So there is interaction between genetic variants and in the androgen receptor gene, as well as the other genes in that pathway. So sometimes you can have a milder phenotype. So that is why I said sometimes when you go into family, you may have a male who is married but is subfertile. You may be able to find that. So it's not a phenotypically female, but it's a male. So therefore, you have to ask for both. If you're looking at the family history, ask for both the subfertility history as well as the primary amenorrhea history, and then the picture would emerge. Okay. Moving on. So in this group, we also talked about the Mullerian agenesis. They are, of course, we're talking about this rare syndrome called Mayer-Rupsky-Hauser syndrome. Which accounts for about 50% of primary amenorrhea, have normal breasts, normal secondary sexual hair development. So they are basically normal looking external. They have normal looking female external genitalia. And if you look at their testosterone level, those will also be normal female level. But then there is absent uterus and upper vagina. If you do a ultrasound scan, and then they will have a normal uterus. Do karyotype 46XX. But you can also see that some of them will also have other abnormalities like renal, skeletal, and middle ear abnormalities. So that's what you see in Mullerian agenesis. We see when we karyotype, there are lots of people. So 50% in that group is a lot, right? Lots of women who have a normal karyotype, and in that case, you are dealing with Mullerian agenesis. Okay. Moving on, what are other possibilities? Absent breast and absent uterus. These are the other possibilities. These are very, very rare. If you have a woman who is masculinized or virilized, you may wonder why. And one possibility is the translocation of a SRY gene onto the X chromosome. So how can that occur? So you know there's recombination in meiosis. So in the father, when there is recombination occurring, the SRY gene from the Y chromosome can get translocated to the X chromosome. How can that occur? How can the SRY gene here, which is here, get translocated there? It occurs due to this reason. This is the X chromosome. This is the Y chromosome. There's exchange of material in meiosis during these two regions of the X and Y chromosomes. These regions are called pseudo-autosomal regions of these two chromosomes. And the SRY gene is just next under that pseudo-autosomal region. Okay, SRY gene is just under that pseudo-autosomal region. And when this is kind of exchanging material, this can also go with that. So when that happens, it gets translocated, and we can demonstrate it. We can do two types of tests, fluorescence in situ hybridization or PCR. When we do fluorescence in situ hybridization, you can put a fluorescence label probe for the SRY gene. So you can see here, this is a big X chromosome, and just under that pseudo-autosomal region, you can see the lighting up of the fluorescence showing the SRY gene, showing that it has got translocated into this person. So you can demonstrate that these people have got the translocation. Generally, we don't do this test we do a PCR test where we just PCR and detect in this person the presence of the SRY gene. That's how we do that. Okay. Then, of course, as we also said earlier, there could be the XY females who are under masculinized males that could occur that way. So all of these phenotypes are there. So remember the spectrum and also what could be there. Now there is also this concept of a pseudo-hermaphrodite. You know? What is pseudo-hermaphrodite? A pseudo-hermaphrodite is a person whose genetic sex is masked by a phenotypic appearance which is of the other sex. So here you are. There's the male hermaphrodite. So you go by the genetic sex. So 46XY is male. So male hermaphrodites. They have testes, and they are the ones, because of a mutation in the androgen receptor gene, get androgen insensitivity syndrome. Then you have the female or pseudo-hermaphrodites. You have the 46XX. They have ovaries, and they get they can be virilized, and that gave you the translocation as one example. But a more common example is congenital adrenal hyperplasia. That congenital adrenal hyperplasia is a more common example of that. What's a true hermaphrodite? True hermaphrodite would be an individual with characteristics of both sex. And how do you determine that? So to do that, you have to do a testicular biopsy, which will show that testis has both ovarian tissue and testicular tissue. When you do the karyotype of these people, you will find that they are 46XX most of the time. Most of the time, they are 46XX. So the complete genetic understanding of how this is caused is not known. We don't know about it. Most of them have ambiguous or female external genitalia. So they belong to this group of ovotesticular disorders of sexual development. All right, so the final message is, when we get these conditions, women presenting with primary amenorrhea, we need to get to the bottom of the genetic etiology. For that, you need to start doing genetic investigation. At the very least, begin with the karyotype. And when we check them, you will find. So if you are interested in, now at almost coming to the final year, if you are interested, just Google, go to PubMed and look at the disorders which are seen in our population, which we have published. You can see all kinds of different karyotypes emerge. We have tested, many hundreds or thousands of people. So all kinds of them emerge. And you can see these are there, out there in the patient populations that you are seeing. So you must do genetic investigation. And again, the true hermaphrodites as well, patients with ovotesticular disorders of sexual development. True hermaphrodites are also there in our population. So we need to investigate these people and reject them. Okay. So I hope in the past 40 minutes or so, I have taken you through the various underlying genetic etiologies, which would eventually manifest mainly as primary amenorrhea in women, but also gave you a little bit of a flavor about other phenotypes that could be there, so that when you are seeing patients in the clinical setting, if you see something which is not so very obvious, you can start thinking a little bit laterally as well. Okay. So with that, that's the end. If you have any questions, I'll be happy to answer them. Any questions? Questions? That confuse everybody. Have you seen any patients with primary amenorrhea in your appointments? Where will you see patients with primary amenorrhea? Would they be in the ward or somewhere else?
In the clinics also, sir, we can see.
Most of them would be in the clinics and be investigated. Unless, of course, they are admitted to the ward. Why would they be admitted to the ward? For which investigation out of the investigations that I mentioned?
Laparotomy, sir.
Laparoscopy.
Laparoscopy.
Yeah. So for laparoscopy, they may be admitted. Otherwise, generally, these patients would be seen in outpatient setting. No? They will not be in a ward. So therefore, important to which outpatient? Which clinic? That also you have to know. Which clinic would you see there? Maybe in a gynecology clinic, or if your hospital, wherever you are, has a special clinic for fertility, subfertility, you may see them in a subfertility clinic. One of those, gynae clinic or the subfertility clinic. Any questions? Any other questions?